Hello everyone, this is our presentation on the adrenaline signaling pathway by Jake Selecky and Ian White. So to start off, we are looking at the lipid bilayer. So the ligand, which is the signaling molecule, which is called adrenaline, is going to eventually bind with the G protein coupled receptor. It's important to note that the receptor for the adrenaline molecule is located on the surface of the lipid bilayer. Because adrenaline is hydrophilic, it's not able to cross the bilayer. So in order for it to bind, it's going to have to be on the external part of the bilayer. And also there's the G protein, which consists of alpha, beta, and gamma subunits. And there's a molecule of GDP attached to the alpha subunit. And this is the protein that's eventually going to bind with the G protein coupled receptor. So the first step includes adrenaline binding to the G protein coupled receptor, and what this does is it activates this receptor, and now that it's activated, it's able to bind with the G protein. So now all three of the substances that we've looked at are now all combined, and since the G receptor protein is activated, it's able to kick off a molecule of GDP from the alpha subunit and replace it with GTP. And now that alpha subunit and GTP are binded, they separate from each other, and they leave the G receptor protein and the beta and gamma subunits. And now it's going to start looking for an inactive adenylyl cyclase because the GTP is able to bind with the right shape and charge of the adenylyl cyclase. And once they bind, this activates adenylyl cyclase. And what activated adenylyl cyclase can do is convert ATP into a derivative of ATP called CAMP, which is called cyclical adenosine monophosphate, something like that. And what these CAMP molecules can do is they can bind to inactive kinases. And you might wonder what kinases can do. Well, once CAMP binds to them, the kinases now become activated. And kinases can break down ATP into a phosphate group and a molecule of ADP, because that's what makes up ATP. And phosphate groups are really important in activating proteins because they change the shape and order of the amino acids, and this causes the function of the protein to change. And this is the main kind of effect of the G protein coupled receptor from adrenaline. And this process is called phosphorylation, adding a phosphate group to a target protein. And so at the very end, the target protein is going to cause a response. And so the main responses that especially come from adrenaline is that the heart is going to beat faster and the lungs, airways are going to open up. And so the main goal of this is to get more oxygen throughout the bloodstream, which is a response of the fight or flight signal that is received from surroundings when something scary or dangerous happens. Now, it's important to note that obviously this adrenaline cannot last forever, and this is visible. Like if someone gets hurt, the adrenaline is going to kick in, but after a while, it's going to eventually wear off. And so what eventually happens is the adrenaline stops binding with the G protein coupled receptor, and then as a result, the GTP becomes GDP, and because this is now a different molecule, adenylyl cyclase becomes inactivated. Now, it's important to see that everything comes back to what originally started. We still have adrenaline floating, and we still have all of the subunits of the G protein. So this process could be replicated very easily if there were another stimulus in the environment. Now, there's still some CAMP molecules that would be, even with the adrenaline not binded to the receptor anymore, there still would be some molecules floating around. So there's some enzymes that are constantly breaking down these CAMP molecules into AMP molecules. And because there's no more CAMP, no more kinases are going to be activated, and therefore no more proteins are going to be activated. So also some of the activated kinases that would otherwise still be floating around are broken down by phosphatase. And so kind of like what I was saying earlier, since the phosphate groups are what activate proteins, since this enzyme breaks down the phosphate groups and separates it from the proteins, the kinases become inactivated, and leaves you with just a normal functioning protein, not heightened like a quicker beating heart cell or lung cell. And so that's all that there is to note. The main things that are unique about the G protein coupled receptor and adrenaline is that there's a secondary messenger in the form of CAMP, which amplifies the original response. So there's many advantages to using a G protein coupled receptor. The biggest one is that hydrophilic molecules are able to be used because since they're not able to cross the lipid bilayer, something's able to, the domain of the G protein coupled receptor reaches out beyond the lipid bilayer and is able to receive the adrenaline. And another benefit is that it involves secondary messengers, which means that a very small amount of adrenaline is able to result in a super big response very quickly, which is really necessary throughout the body. Now, a couple downsides of it is that the response really doesn't last for that long, and it's just super temporary. And thank you very much for listening.